One of the joys of my job is that I'm a sociologist by training, but I get to always be a learner. And so I get to work with faculty like Mark Johnson across the disciplines, sometimes also sit in on their classes and learn from them.
I'm Mark Johnson, faculty member in the department that's called MCB. And MCB stands for Molecular Biology, Cell Biology, and Biochemistry. I'm a geneticist who studies plant reproduction, and I teach genetics here at Brown. And I also teach a class called Inquiry in Plant Biology that is an example of a collaborative scholarly experience, and Mary is about to introduce you to what that means. I also am involved in an effort at Brown to innovate, particularly in science teaching. And that effort is funded by the Howard Hughes Medical Institute. So over the last, I guess about 10 years now, I've been involved in trying to change the way things operate here for STEM, particularly science education, to make it more accessible, to make it more hands-on. And this CoEX effort is one of the things that I can point to and say was a really positive outcome of that work. So yeah, it's really nice to meet all of you today. Welcome to Brown, and I'm glad we have such nice weather for this.
I'm going to give you a glimpse of his class, his CoEX class, so that you can experience it as a learner. That's going to be a lot of fun. But first, because it all boils down to, in our jobs, about learning, we want to hear about your experiences as learners. Picture that time in your mind, rewind the mental tape, and think about for a minute, what exactly was happening at that time?
This could have to do with something that you had to learn for work or in any context. Like, think about what got you really excited about learning something new.
In about 2 minutes, or what are some of the commonalities in those experiences when you are highly engaged with the experience? For those of you sitting alone, we encourage you to get the blood flow moving and find a partner.
We're Elizabeth and David Hayes from Hillsborough, North Carolina, and our commonality is that there were other people in conversation with us.
I'm Vidula. I'm from Ohio, and this is my dad. He's also from Ohio. And we were talking about how we were both able to go at our own pace and work at learning something new within reach of what we were capable of doing.
Okay, so self-directed and just a little bit of a stretch,
Yeah.
but it is achievable. Yeah, thank you, Vidula.
I'm from Oregon, and the couple of things that first popped into mind were classes I had where I had a sort of a personal connection with a professor.
I'm Dennis. This is my daughter Molly. I'm gonna let her do all the talking though.
Hi, so we both found that we had similar learning experiences because we both were, I guess, like discovering the ins and out of like a certain program we were using to produce something artistic, and it was interesting because we both found, I guess, it interesting to learn about the technical processes behind trying to create something.
So the discovery or the process of the creative process was what was engaging for
Yeah.
you. Yeah. Thanks, Molly.
I just want to add also, we also shared in common that once you're learning this kind of technical process, you also find ways to kind of bend the rules in that process and have your own style.
I'm Pablo, this is Barbara, we're from Mexico. What's common from our experiences is that we both wanted to, we were aspiring to do something better in the future. So that's what we were learning, the different stuff. She's an architect. It was a software program that was very tough. For me, it was learning the German language.
I think the commonality we found in terms of when we get engaged with learning something new is just the excitement of learning something new and knowing that we're going to be walking out of that experience with an additional skill.
Yep. So this is a biology course. We also have one in engineering as well, advanced fluid mechanics. But we also have these across the disciplines. So here's a new one offered last year on virtual reality. It was co-offered between cognitive linguistic and psychological sciences and Italian studies. So these can be interdisciplinary as well. And here was a really interesting one offered in Africana Studies. So a beloved professor of Africana Studies sadly passed away last year. But if you can take a look at his office, it's a treasure trove of important information that needs to be archived. So now I'm going to turn things over to Mark again, and he's going to give you a little bit of a glimpse of what it's like to be in a CoEX course. based on his experience teaching inquiry in plant biology.
So this is a course that I've taught with a partner in my department named Allison DeLong over many years. And that process then leads to development of seeds. So the ovule turns into a seed. And without that fertilization process, the fruit, like in the case of a tomato, also won't begin to develop. So this process is required to make a kernel of wheat, a grain of rice, a soybean, the tomatoes that you eat. And it's a really amazing cellular process because this one cell grows from here to here. So this is just it's a simulation, a cartoon version of what the pollen grain does. So it lands, it hydrates, it makes this projection, it grows at rates of millimeters per hour, which is really fast for a cell. It responds to attractants and grows out to the ovule and explodes and delivers these 2 sperm cells that fertilize the egg and the central cell to create seeds. So this is what a pollen grain looks like. Well, yes and no. So irrigation can help because the combination of drought and heat stress is even worse. But heat stress alone is sufficient to prevent fertilization. Yeah, so that compounds the challenge if you add drought. But this is an experiment that was done in my lab, actually by a student who went to Brown and then worked in my lab for a few years and then decided he wanted to do his PhD. So he is a Brown alumnus both as an undergraduate and a PhD student.
The only difference between the green ones and the red ones was that the green ones first were growing through the floral tissue before they got onto the surface of the medium. The red ones didn't. We took them straight out of the anther and put them near the ovules. So yes, the model is that there's something about growing through the floral tissue that activates the pollen tube and enables it to respond to the signal that the ovule is giving off that attracts it to it. Yeah. And so these experiments that were done a while ago led to that hypothesis. And then later, proteins called receptors were discovered that the pollen tubes express that allow it to respond to other proteins that the ovule is secreting that pull the pollen tube to it. And then what we learned was that the expression of those receptors is controlled by growth through the flower tissue. Yeah. So nice job. You were all just did the work of scientists for the last 15 minutes. This is how we would start our class, and then students work in groups to design their own experiments to follow up on the observations that we might start with. So in the context of our course, what we might do is now design an experiment that would test how temperature affects this process. So at this point, we might say, okay, now work together. We've given you an experimental setup. How can you manipulate this to ask, how does temperature affect the ability of pollen tubes to find their targets? Okay. So do you want to work on that? Or do you have questions for me? Yeah, so this is a really good control experiment. So another hypothesis is, well, there might be something genetically different about the red ones. The cartoon version, there's only one pollen grain that's been put on the stigma. And, here it comes. So matches equation. So now it's growing down. And the question is, what makes it, I think it ends up at this one. What controls where the pollen tube ends up in this field of ovules? Okay, so each of these pink cells, or they're depicted as pink in this cartoon, that's called a synergid cell, which is right next to the egg cell. That synergid cell is secreting a protein that was cleverly named by a colleague of mine in Japan, lure proteins, L-U-R-E, because they lure the pollen tube. So each of these cells is secreting kind of a field of lure proteins out into the environment, and they kind of ooze down along this stalk, and then they can begin to interact with the pollen tubes as they grow through this central tissue. So the current model is that if you do the experiment with one pollen tube, it'll go to whichever ovule it first gets within the range of that floor signal. Now, if you do it with hundreds of pollen, they're all sort of racing down. And the first one that gets in range of an ovule that's secreting the spore will go to that one. But if you do experiments with like varying amounts of pollen, you find that they don't always go to the first one, the top one. And it kind of, I think, and this is what we don't know, it depends on where the pollen tube is growing in this space. So if it gets close to one, it will go to that one. But these attractants work at a specific range. And I think the best hypothesis now is that that's what determines which ovule gets fertilized first. Yeah. But that's exactly the kind of question that can launch like a whole PhD project. Yeah. Let alone keep a group of 20 students very engaged for a semester-long course.
So good question.
Yeah.
Other questions? Bring it to the back.
I wonder if there is anything we can learn from the plant reproductive system dynamics that would apply to other animals or human beings.
Yeah. So this kind of cellular homing process is not limited to inside of flowers. In fact, the way that our axons find where they should grow within our bodies as we develop, early on in life when our whole nervous system is being wired up, the cells that are involved in that process work in a very similar way. So they grow by the same mechanism where they're extending at their tips, and they use receptors and signaling molecules to guide them to their right destinations. Now, like to the practical thing, the way this system evolved in plants is, we now know, completely different from how that similar system evolved in animals. And so the molecules that are involved are different. So in that case, we can learn sort of general principles, but not specific molecules. But another project in our lab is focused on what happens after the sperm are released.
HAP2 that's expressed by the sperm and is essential for the sperm cell to fuse with the egg cell. So like that fusion of two cells is actually a complex process because the cells are surrounded by this membrane that normally would get like water trapped between it, and then the membranes can't merge. But when a sperm and an egg come together, those two membranes have to merge to form the zygote. Believe it or not, in humans, we don't know what the mechanism that makes a sperm and an egg plasma membrane fuse. We don't know it for any, we don't know that mechanism for any vertebrate, but we do know how it works in plants by studying this system. And the molecule that we discovered in the plant system is very broadly conserved, meaning we can find it in some animals. We can find it in just about every plant by looking in the genome of those organisms. But it's still in the human genome, we can't find a protein that has a similar sequence as this protein. But we're using what we're learning about this protein in plants to search for the human protein that might be doing that function that's obviously critical for reproduction. Yeah. So that's, those are just two examples that are close to my work, where work in plants is obviously important for agriculture, but it can also be applied to animal systems.
Can students find these CoEX classes in other disciplines than the biology or any other science classes?
Yeah, thank you for that question. So the answer is yes. They are broadly available. Mary mentioned a few at the very beginning that are in a few different departments. So just to review, there was one in Africana Studies. There is one in Cognitive Science, Linguistics and Psychological Sciences. There's one in Engineering. There are maybe 3 or 4 in Biology. And let's see, there's one in Chemistry. And then the goal is to really expand this out. And Mary and the Sheridan Center are actively involved in doing that now.
And so next term, there are actually 12 being offered across the disciplines, also sociology as well.
Yeah. And it's interesting, like, we're involved in working with colleagues across the university to encourage this kind of thing. depending on the style of research that's done or the type of scholarship or even in a creative arts discipline, people have, they might need to think and learn a little bit about how to do this. But almost always we find that people really get excited about it because it gives you an opportunity to teach about something that you really care about. And it goes back to that characteristic of learning. Like for the students, it also gives them an opportunity to participate with somebody who really cares about what they're doing. Yeah.
Well, thanks, Mark, and thanks to all of you for joining us. We'll stay after for a little bit if you have other questions.
There is one. Do you want to ask your question?
Sure. Class would have been taught before if it were not CoEX.
Yeah. Okay. So the question was, how would my class have been taught before when it was not CoEX? So yeah. So I arrived here in let's say 2004, and then like a year later, they told me I was going to teach this course, and I was like, all right, what are we going to do in the lab? And so, I like having pollen tubes. I love pollen tubes. So we're going to do something with pollen tubes. So, but basically what I did was have them grow pollen tubes, and then I told them a variable that they could change that would affect pollen tube growth, and I just asked them to see if they could measure a difference. So the difference was, like, I gave them a recipe. I said, here's a variable that you could change it, and see what you get. And they learned things, and they learned how to make measurements. They learned how to analyze results. That was all good. But the part that you did today, where you got to ask the questions and design the experiments, we weren't doing that. So it requires a little bit more background to get the students thinking and designing the experiments, and takes a little bit more time to get started, but then ultimately what they do is much more interesting. Yeah.
Fantastic.
All right.
Okay, well, thank you.
Thanks, everybody.
Yep.